Today we're going to be talking about the process of DNA replication. And before we do, we need to talk about why DNA replication exists. As we know, DNA is incredibly important to all organisms because it contains the instructions for making proteins that are going to coordinate the activities and basically regulate the activities of that organism. So, in order to do that, and in order to do that for future generations, and in order to do that for new cells, the DNA must be replicated. Before an organism can produce a new cell, it must first replicate the DNA, because we can't simply take a cell and divide it in half. If we do, then we split the DNA in half. Now our instructions are cut in half and the cell will no longer function. So before a cell ever thinks about dividing, it first has to replicate its DNA. And it doesn't matter if we're talking about, creating a new organism, or if we're talking about just making a new cell. Before any cell undergoes mitosis, the first thing it's going to have to do is replicate that DNA so that we have two copies and one can go to each of the new cells created. So DNA replication is going to be happening in our cells all the time. Like our cells, we're constantly replacing cells that are damaged, that are worn out, for whatever reason. Anytime we're replacing a cell or making a new cell, we're going to need to undergo this process. A DNA molecule is double-stranded, so the first process that we're going to have to go through in order to replicate it is to separate the two strands, right? So we have two strands of DNA in our double helix model. We're going to have to separate these strands. So we're unraveling, and we call this a replication fork, where the two strands have separated from one another, okay? And so we're going to use one of these strands, we're actually going to use both of these strands as complementary strands, or we're going to use them as templates, right? So a template is kind of like a blank document that's kind of like halfway filled out for us. Like if you're going to write a letter to someone, you may use a template, and it says, put the dear part here and put sincerely down here. So that's a template. So what we mean by that in DNA is these base pairs are going to be used so that we can add the correct new base pairs. So if we zoom in right here on this strand, let's say it reads A, T, T, G, okay? Following the rules of base pairing, what we would need to do is add a T here, and an A here, and an A here, and a C here. Meanwhile, on the other side, if we were to zoom in on the other side of this strand, okay, so this was our original strand, this is what we added. So I'll put original, okay, and this is what we added. If we were looking at the other side, originally when it was complementary, it would have read T, A, C, right? Because T goes with A goes with T, A goes with T, and C goes with G. So now on this side, when we add the new base pairs, we're adding an A, T, and a G. And when we look at these two now molecules of DNA, they are identical. They each have one original strand and one new strand. One original strand and one new strand. So that original strand served as the template, and then we just follow the rules of base pairing to ensure that when we are done, we have two identical copies of the DNA, because that's what we're going to have, right? In order for the cell to function, in order for the organism to maintain and continue to be that same organism, those strands must be identical when this process is complete. So this is known as the semi-conservative model, as each molecule now contains a parent or old strand and a new strand of DNA. Okay, so what they mean by semi-conservative is we conserved part of the original strand, right, or part of the original molecule, and then we have a new strand. So remember, each DNA molecule consists of two strands. And if we're talking about like a molecule that was recently replicated, it would be the original and then the new. And then over on this side, the original and the new. So that's the semi-conservative model. And again, we're always going to be following the rules of base pairing, where adenine pairs with thymine and cytosine pairs with guanine. You can't get bonds between thymine and cytosine, so you don't have to worry about that happening. There's a lot of fail-safes built into DNA replication. like base pairing, that's a fail safe, right? So you can't get the wrong base pairs together. Also, once this process is over, then you have basically a molecule that comes through and proofreads it, and it can even make corrections. So we want to make sure that we get this right, because if we change our DNA, then we're potentially changing, the organism's proteins and potentially changing traits. So it's very important that the process runs along seamlessly and that it gets done without any mistakes. So the way we're going to do this, and we'll get a little more technical into it, is it begins with DNA helicase. And DNA helicase is an enzyme that's going to start untwisting our two strands from one another. Okay? It's going to untwist those two strands and separate the hydrogen bonds. So those hydrogen bonds that hold the nucleotides together, we're going to break them apart, and now we have two separate strands. Okay? The strands have specific sequences that let the enzyme know where to bind, and that is called the origin of replication. So that's where this whole process is going to start. This forms what is referred to or called the replication bubble. At the end of each bubble, the strands are split, and that forms our replication fork. Okay, so you have your bubbles there, and then, like I said, at the end of those bubbles, you are going to have your fork, just like a fork in the road where it goes two different directions. You're going to have your forks there at the end of each bubble. Once split, replication begins in both directions on both strands simultaneously. So both strands are going to be running at the same time. DNA polymerase is an enzyme that is going to bind to each strand, and it starts binding complementary bases, creating the new daughter strand. So, we have our new strand over here, we have our new strand over here. DNA polymerase is going to start putting those base pairs where they need to go. Again, scientists not being super creative, smart bunch, never known for their creativity. DNA polymerase is an enzyme, ASE, that turns DNA into a polymer. It is linking together the base pairs. So that's a perfect name for that enzyme, because it is turning DNA into a polymer. It adds the sugar and phosphate backbone that join the base pairs as well. So DNA polymerase takes care of that process. The DNA strands themselves are anti-parallel. So they're running anti-parallel to one another, and the DNA polymerase can only add continuously at the 3' end. So this is where it gets, especially if you've ever seen a video of this or you've seen it in action, it gets a little tricky. So one of them, it's just going to build continuously in that direction. So that strand is just built continuously. The other strand, moving in the opposite direction, is going to be built in what are called Okazaki fragments. And Okazaki fragments are named after the scientists that discovered them. They basically have breaks in between them. And so you have a continuous strand moving in one direction, and then you have the discontinuous strand, which is being built in fragments moving in another. Another enzyme called DNA ligase comes in to link the pieces together to make a single strand. So those fragments, it's going to make them all together. These enzymes, as I mentioned before, these are the ones that are going to do the work of proofreading and repairing the DNA strand. So like I said, we've got to make sure we get it right. So DNA ligase is going to not only proofread to make sure that it's correct, but also make any necessary repairs to the DNA strand. Once it has been replicated, they make another pass over the strand and remove incorrect base pairs and put in the correct complements. So like I said, there's all kinds of fail-safes built into this process to ensure the end product is what we want and not something that we can't use. In eukaryotic cells, telomeres are added to the ends of the strands as they are straight instead of circular. So pieces at the ends of the strands get cut off. Telomerase adds a poly A tail to the ends, which makes it less likely that genes will be cut off or damaged. So it's kind of a cap there that protects the end of that strand. So once that's all done, once all these enzymes have done their job, what we have and what we end up with are two identical molecules of DNA. One will go to one cell and one will go to another cell, and that ensures each daughter cell has their own copy of DNA, and as soon as that cell is created, it is capable of doing all the things that it needs to do because it has the instructions to do so. thanks to the process of DNA replication. I'll see you next time.